Welcome back to the deep dive. Stop for a moment and consider the invisible world all around us and even within us.
Mhm.
We're talking about microbes, those microscopic architects of health and disease. For centuries, our understanding, well, it was limited, wasn't it? Just what we could see through a microscope.
Right, like trying to understand an entire library by just looking at the book covers. Very limited view.
Exactly, but what if you had a superpowered microscope, not really for seeing, but for reading the very blueprint of life itself, the genetic code of every microbe?
That's the power we're talking about.
That's the incredible power of infectious disease genomics. Today, we're taking a deep dive into how this cutting edge science is completely transforming our understanding of, well, everything from a common cold to global pandemics.
It really is a fundamental shift, isn't it? We're moving from just, observing the effects of microbes to directly reading their underlying instructions.
The instructions themselves.
Yes. And our mission for you in this deep dive is to really gain a master level understanding, just like the elite professionals in this field. We want you to grasp how these technologies are reshaping clinical practice, public health, our very fight against infection.
We've distilled insights from extensive research, clinical observations, to get right to the core of what's truly important for you to know, so you can walk away feeling completely informed.
That's absolutely the goal.
And our insights today come directly from a comprehensive chapter on microbial genomics and infectious disease. It's packed with groundbreaking examples, the latest scientific literature.
It really is.
Historically, microbiology relied so heavily on visualizing microorganisms, growing cultures in petri dishes, then peering through a microscope.
You grew it, you looked at it, you tried to figure out what it was. That was the process.
But now, genomic advances are providing, well, just unprecedented resolution, allowing us to characterize the entire genetic map that underlies all microbes.
And that illuminates their complex interactions with each other, the environment, human health, in ways we never thought possible before.
When you connect this to the bigger picture, the field of infectious disease genomics really represents a vast, rapidly expanding frontier.
Oh, absolutely. It's moving beyond simply identifying a pathogen to understanding its entire genetic potential. What it can do, how it might evolve, even where it came from,
And that fundamentally transforms clinical practice.
fundamentally.
Okay, before we jump into the revolution itself, let's make sure we're all speaking the same language. We need to cover some fundamental terms. These are absolutely critical if you want to truly master this deep dive. First up, the genome.
Right, the genome. It's quite simply the entire set of heritable genetic material within an organism.
The whole instruction manual.
Exactly. Think of it as the complete comprehensive instruction manual for that microbe. Before genomics, we were often only able to read like a few fragmented pages.
Just bits and pieces.
Yeah, bits and pieces. Now we're reading the entire book, cover to cover, and that gives us an unprecedented level of understanding.
Okay, and to read that incredibly complex book, especially at the scale we need for infectious disease, we rely on next-generation sequencing, or NGS. How's that different from the older ways?
Well, NGS refers to high-throughput sequencing. It's a massively parallelized process. It produces millions, sometimes even billions, of sequences concurrently.
Wow.
It's a revolutionary leap beyond the prior, much slower methods like dye terminator sequencing. It just offers unparalleled speed and scale.
Like reading millions of pages at once instead of one at a time.
That's a great analogy. That's exactly the difference.
Which then brings us logically to whole genome sequencing, or WGS.
Right. WGS is simply the process of determining the full DNA sequence of an organism's entire genome. And it's greatly facilitated by NGS, obviously.
Makes sense.
This technology is precisely what allows us to shift from looking at individual genes, just specific chapters or paragraphs, to analyzing the complete instruction manual of a microbe. It gives us a far more holistic view of its capabilities and, importantly, its vulnerabilities.
Okay, and one more term. It might sound a bit technical, but you said it's crucial for understanding how we piece these genomes together, especially for new discoveries. CONTIG.
Yes, CONTIG. CONTIG is basically a DNA sequence representing a continuous fragment of a genome. Imagine you're trying to piece together a shredded document.
Okay.
You get lots of small overlapping scraps, right? A contig is one of the longer continuous stretches you can build by matching and joining those overlapping fragments.
I see.
And this is especially relevant for what we call de novo assembly. That means when we sequence a new, unknown microbe, where we don't have a pre-existing reference genome to compare it to.
Right, starting from scratch.
Exactly. Contigs are those essential building blocks that help us reconstruct the full, previously unknown genome.
Okay, great. With those foundational terms now firmly in mind, let's jump into our first major section. This is the foundational shift from phenotypes to genotypes in diagnostics. This is where we really start to see the practical impact, right?
Absolutely. This is where the rubber meets the road, clinically speaking. Traditionally, the clinical microbiology lab, well, it had some core goals.
Like what?
Establish if a pathogen is actually present in a sample, identify it, and then, if possible, provide information for clinical management. Things like antibiotic susceptibility or maybe the presence of virulence factors.
And for generations, their approach was largely phenotypic, based on observable traits, literally what you could see or measure in the lab.
That's exactly right. It meant growth-based assays, biochemical testing, sometimes just grouping bacteria by how they looked under a microscope or what nutrients they needed to grow, or if they could catalyze certain chemical reactions.
And antibiotic susceptibility, you mentioned.
Yeah, that was determined purely by seeing if bacteria could physically grow in the presence of an antibiotic in a test tube or on a plate. Very direct observation.
But the limitations there seem pretty obvious, especially the time it takes.
Well, absolutely. The inherent limitations were significant, primarily the delays. You're waiting for microbial growth. That can take 1 to 2 days just for identification, then maybe another 2 to 3 days for susceptibility results.
And for some bugs, even longer.
Right. For slow growers, like Mycobacterium tuberculosis that cause a TB, this could stretch into weeks.
Weeks. Wow. And that delay has huge implications for treatment, right? Forces doctors to guess.
Precisely. It has huge implications for what we call empirical antibiotic use starting treatment before you have a definitive diagnosis. It often leads to using broad-spectrum antibiotics.
Not a shotgun approach.
Exactly. And those choices might be unnecessary, or worse, incorrect. And this contributes directly to the rise of antibiotic resistance globally. It's a major problem fueled by diagnostic delays.
So, if traditional methods were like guessing or waiting, what's the game-changing promise of this sequencing revolution? How does reading the instructions change the approach?
Well, the core insight, and this is key for you to grasp that mastery level, is that easily accessing complete pathogen genomes allows for a systematic understanding of the genetic basis for those observable phenotypes.
Ah, so you know why it behaves that way.
Exactly. No more guessing why a bug acts a certain way. We can read its instructions and know its potential. And this translates directly to improved outcomes.
How so?
Nucleic acid-based diagnostics, built on this genomic information, promise significant improvements in, well, speed, sensitivity, specificity, and just the sheer breadth of information compared to traditional methods.
So better on almost every metric.
Pretty much.
Yeah.
And these technologies aren't just theoretical. They are already delivering on this promise. They're bridging the gap between clinical labs and research labs, which used to operate, quite separately.
That sounds incredibly powerful. So how exactly have these genetic approaches helped us overcome those historical limitations? Let's talk about those unculturable pathogens, the bugs that just refuse to grow in the lab. What did we do before genomics for those?
Right, the unculturables. Before genomic methods, diagnosing them relied primarily on two main things, serology or antigen detection.
Okay, what are those?
Serology measures the host's immune response looking for antibodies, for instance, to pathogen exposure. Antigen detection looks for specific components, like proteins, of the pathogen itself.
But both of those have significant downsides, don't they? Serology, for example. It sounds slow.
It often is. It frequently only allows for retrospective diagnosis. You need both an acute sample, taken when the patient is sick, and a convalescent sample, taken weeks later, to see a change in antibody levels. That takes time.
Not helpful for immediate treatment decisions.
Not at all. And for chronic infections, distinguishing between an active infection and a latent one, or even just a past exposure, can be basically impossible with serology alone.
So you might have antibodies, but not an active infection.
Exactly. You might have antibodies to Lyme disease, for example, but that doesn't mean you have an active infection needing antibiotics right now. It could just reflect a prior exposure years ago. Similarly, staging syphilis by serology is really complex, requires multiple tests to interpret properly.
Okay, so serology has issues. What about antigen detection?
Well, antigen detection wasn't much better, frankly. It offered limited clinical information, often had suboptimal sensitivity and specificity, meaning false negatives or positives, and could still involve long delays.
And couldn't tell you much beyond it might be there.
Pretty much. Crucially, it couldn't characterize pathogens beyond just identifying potential past exposure. And it's only validated for a limited set of infections, usually targeting structural things like viral envelopes or bacterial and fungal cell surface markers.
This really sets the stage for the truly revolutionary breakthrough. Nucleic acid amplification tests, or NEMATs. What exactly are they? How do they work?
Okay, NETs. A NET is essentially a biochemical assay. It evaluates for the presence of a particular string of nucleic acids that could be DNA or RNA by amplifying it.
Making lots of copies.
Exactly, making millions or billions of copies. The most common method, one you probably heard of, is PCR, polymerase chain reaction. It takes a tiny, almost undetectable amount of a pathogen's genetic material and copies it over and over until it becomes easily detectable.
Are there other methods besides PCR?
Yes, there are others, like ligase chain reactions, but PCR is definitely the most widespread.
And NETs have just become standard practice for so many viral infections now: HIV, hepatitis C, influenza. How did this transform our understanding, say, for something like HIV?
Oh, for HIV, it was a complete game changer. Before sensitive gnats, our understanding of HIV's natural course was incredibly limited, almost misleading.
How so?
Well, back in the early days of the AIDS pandemic, the initial tests relied on detecting a viral protein called P24 antigen. But this antigen was fleeting. It would disappear for years after the acute infection phase.
So it looked like the virus went away.
It made it seem like the virus went silent, yeah. Without sensitive viral load tests, which eventually could detect as few as 20 copies of viral RNA per milliliter of blood, the role of early treatment was really uncertain. People didn't know if treating early mattered.
It's remarkable. So the advent of these more sensitive viral load tests completely changed the picture, showing viremia, the virus, being present in the blood as a near universal feature of HIV infection.
Exactly. And because the older phenotypic assays, which measured drug resistance by actually trying to grow the virus in the presence of drugs, were so cumbersome and slow,
Right.
genotypic antiviral resistance testing became the standard of care, even before starting therapy in developed countries. You sequence the virus first.
So you could tailor treatment right from the get-go.
Precisely. It allowed for truly tailored treatment plans right from the start. These genomic developments, combined with therapies that were becoming less toxic, really shaped the policies towards earlier introduction of antiretroviral therapy. Massive impact.
And we saw this play out on a huge global scale with COVID-19 and SARS-CoV-2.
We absolutely did. RT-PCR assays, that's reverse transcription PCR, specifically for RNA viruses like coronaviruses, became the core method for detecting acute phase infection.
The main test everyone was getting.
Exactly. It formed a critical component of both the clinical and the public health response. It actually represented the largest implementation of a single molecular infectious disease assay in history. A truly global effort built on that NAT technology.
But gnats aren't just for viruses, are they? They're also key for certain bacterial infections.
That's right. They are the diagnostic tests of choice for what we call fastidious bacteria, meaning bacteria that are difficult or slow to grow in the lab.
Like which ones?
Common examples are Neisseria gonorrhea and Chlamydia trachomatis, the causes of gonorrhea and chlamydia. Also Clostridium difficile, or C. diff.
And for C. diff, can gnats tell you more than just it's present?
Yes, and this is important. For C. difficile, gnats can provide clinically relevant information beyond just identification. They can detect the presence of the genes for its cytotoxins, like toxins A and B.
Which tells you if it's likely causing the diarrhea.
Exactly. It helps distinguish harmful, toxin-producing strains from harmless colonizers. Some gnats can even detect markers for hypervirulence, like the NAP1 strain, which is a particularly aggressive variant known for causing severe outbreaks.
That's a clear win for diagnostics. But does relying so heavily on these specific genetic signatures ever create new challenges? I mean, what happens if the target sequence changes?
That's a crucial point, and it highlights the need for ongoing monitoring. The C. trachomatis variant that emerged in Sweden is a perfect example.
What happened there?
It had a deletion, a missing piece in its genome, right in the region that several commercial gnats were designed to detect.
So the tests missed it.
The tests completely missed it. This led to its widespread prevalence in some areas because it simply wasn't being detected and therefore wasn't being treated effectively.
That's concerning.
It's a stark reminder that even with these incredibly advanced tools, careful development and critically continuous monitoring of molecular diagnostics are absolutely essential because pathogens evolve. We have to keep checking that our tests still work against the circulating strains.
Okay, so looking ahead now towards the future of microbial diagnostics, what's the ultimate goal? What's the North Star here?
The drive, overwhelmingly, is for rapid, real-time pathogen identification. And ideally, that comes with antibiotic susceptibility information included. right away.
Instant diagnosis and resistance profile.
That's the dream. This has the potential to profoundly transform infectious disease management. Think about the impact on antibiotic stewardship, on reducing mortality risk in critically ill patients, even on hospital admissions and how long people stay,
So it moves from reactive, delayed decisions.
to proactive,
Yeah.
immediate, and precise interventions. That's the core insight here, the shift we want you, the listener, to really internalize for that deep understanding.
And it's a massive shift for public health, too, not just individual patient care.
Huge. We're already seeing public health agencies like those in the US and UK shifting away from older, less granular methods like pulse field gel electrophoresis, PFGE,
Right.
towards routine whole genome sequencing for tracking things like foodborne pathogens or other microbes like mycobacteria and gonorrhea.
And international efforts are doing the same.
Absolutely. International efforts tracking major outbreaks like Ebola, Zika, influenza, and of course SARS-CoV-2 are relying heavily on genomics for improved interventions, better surveillance, and even guiding vaccine development.
A key question then is, what's making these powerful technologies more accessible? What's lowering the barriers to adoption, especially in places that might not have sophisticated labs?
That's a great question. Several things are happening. We're seeing the development of isothermal amplification protocols. These don't need the complex temperature cycling machines of PCR.
Simpler equipment.
Much simpler. And lyophilized reagents basically freeze-dried chemicals that don't require refrigeration or sophisticated instruments. They're stable at room temperature, easier to ship and store, perfect for field use or basic labs.
And cost. Speed?
The cost and speed of sequencing itself continue to fall precipitously. It's remarkable how fast and cheap it's become compared to even 5 or 10 years ago.
Moore's law for sequencing.
Kind of like that, yeah. Plus, automation and miniaturization of sample preparation are reducing both the cost and the specialized expertise needed. And we're seeing improvements in direct sequencing technologies, which aim to bypass some of the complex molecular biology steps altogether, making the whole process potentially simpler and faster.
So putting it all together, what does this mean for the everyday clinician, the person on the front lines? What's the ultimate clinical goal?
The ultimate goal is to provide that clinician with real-time information. Information on whether antibiotics are even indicated, and if they are, precisely which ones will be effective against the specific pathogen causing the infection in that patient.
Personalized, immediate guidance.
Exactly. This enables a far more efficient, targeted deployment of our precious antibiotic arsenal. It helps preserve its effectiveness for the future, reduces side effects, and improves both societal and patient-specific outcomes.
It's almost like you mentioned earlier how rapid troponin assays transformed managing chest pain. Immediate, actionable data.
That's a very good parallel. It's about getting that critical information quickly to make the right decision right away.
Okay, that makes perfect sense. Now, let's shift gears slightly. We've talked about the broad strokes of diagnostics. Let's get more specific. Pinpointing the culprit organism identification and pathogen discovery.
Right. Finding that needle in the haystack, or sometimes, as you said, discovering the haystack itself is something completely new.
So how do we use nucleic acids to actually identify specific organisms? What's the challenge there?
The challenge is finding sequences, stretches of DNA or RNA that are conserved enough within a species so you can detect all its different strains, but also divergent enough between species so you can tell them apart accurately.
Finding that sweet spot.
Exactly. Historically, for bacteria, the go-to target has been the 16S ribosomal RNA gene, or subunit,
Why that one?
because it's highly conserved across almost all bacteria, essential for their function. But it also has variable regions within it that contain enough unique sequence information for identification, down to the genus or sometimes species level.
Can you give an example where 16S was crucial?
A great example was confirming those Mycobacterium chimera infections after cardiothoracic surgery. 16S sequencing helped link cases and eventually recognize a widespread outbreak tied to contaminated equipment.
But you mentioned it has limitations.
It does. While 16S PCR can be done in specialty labs, its sensitivity, especially directly from tissue specimens, can be limited, and sometimes it doesn't give enough resolution to distinguish very closely related species.
So what happens when we need more information than just a general bacterial ID?
That's where the wealth of full sequencing data really comes into play. Now, with more complete genomic information readily available, we can go way beyond just the 16S gene.
Targeting other genes.
Right. We can target other genomic loci that provide additional, often more clinically relevant information. For instance, as we discussed with C. difficile, knowing not just that it's present, but detecting the presence or even the expression of its toxin genes, toxins A and B, directly helps clinicians distinguish those harmless colonizers from the true toxin-producing pathogens causing disease. Same idea for detecting Shiga toxin genes in E. coli.
And this leads into the idea of syndromic panels we see in clinical labs now. What are those exactly, and how do they streamline diagnosis?
Syndromic panels are essentially multiplexed PCR assays,
Multiplexed meaning?
meaning they can simultaneously test for multiple common causes of a specific clinical syndrome, all from a single patient's sample.
Ah,
So you might have a respiratory panel that includes primers, the short starting sequences for PCR targeting influenza, SARS-CoV-2, RSV, various other common respiratory viruses, maybe even some atypical bacteria, all in one test run.
so one swab, lots of answers potentially.
Exactly. The goal is streamlined diagnosis. If you can quickly identify a plausible cause, it offers diagnostic clarity. Of course, it needs to be used judiciously and interpreted within the patient's whole clinical picture. But it's about getting a broad yet specific answer much faster than testing for things one by one.
Okay, these PCR-based assays, syndromic or single, are clearly powerful, but they still often require fairly complex lab equipment, right? Temperature cyclers, specific readers. This raises that important question again. How do we make these powerful nucleic acid diagnostics even more deployable, especially in resource-limited settings or even potentially at the point of care?
This is where some really exciting new technology is emerging, particularly CRISPR-based diagnostics.
Clipper, like the gene editing tool.
The same underlying technology, yes, but repurposed for diagnostics. These tools build on the incredible sensitivity and specificity of certain CRISPR enzymes, like Cas12a and Cas13a, which act as programmable nucleic acid sensors.
How do they work as sensors?
These enzymes can be programmed to recognize very short, specific nucleic acid targets, like a sequence unique to a particular virus or bacterium, with extremely high specificity. When they find their target, they get activated. And this activation triggers a sort of collateral cleavage activity.
Meaning they start cutting other things nearby.
Exactly. They start cutting other nearby nucleic acid molecules non-specifically. And researchers have engineered reporter molecules that release a fluorescent signal when they get cut by this collateral activity.
So target found, enzyme activated, signal lights up.
Precisely. And the key innovation here for deployability is that this can often work with isothermal enzymatic preamplification.
Isothermal means?
Constant temperature. It doesn't need the precise rapid heating and cooling cycles of a PCR machine. Much simpler hardware.
Okay, that's a big deal for field use.
It is. And the reagents can be robust enough to be lyophilized, freeze-dried onto paper strips, making them stable at room temperature without refrigeration. You could imagine simple, potentially handheld devices. These assays are still relatively early in development for widespread clinical use, but they show immense promise for global diagnostics and surveillance, potentially bringing sophisticated molecular testing to places it's never reached before.
That's fascinating. Now, beyond amplification tests like PCR and CRISPR, you mentioned hybridization-based detection methods earlier. How do those fit in?
Right, hybridization. While not yet widely used clinically for primary diagnosis of infection, these techniques, like DNA microarrays, are being developed and used for other purposes, like typing or characterizing pathogens.
How do they work?
They work by having known nucleic acid sequences, probes, attached to a surface, like a microarray chip, or floating in solution. You then add the nucleic acid from the patient sample. If the sample contains a sequence complementary to one of the probes, it will bind or hybridize to it.
And you detect that binding.
Exactly. One potential advantage is that hybridization can be more tolerant of slight mismatches in the sequence compared to the strict requirements of PCR primers. This might allow detection of more diverse isolates within a species.
Any other advantages?
Importantly, basic hybridization doesn't require complex enzymology like PCR, potentially broadening the conditions for their use, maybe even directly on primary clinical specimens without much processing. And because they can work with both DNA and RNA, it opens up the possibility of expression profiling, looking at which genes are actively being transcribed into RNA, which can uncover phenotypic information directly from the nucleic acid content.
Okay. Now, while PCR, CRISPR, and hybridization generally target specific known organisms or sequences, what about the situation where you have absolutely no idea what you're looking for? What's the ultimate unbiased approach to identifying pathogens, or maybe even discovering completely new ones?
That's where metagenomic sequencing comes in. This is really the frontier for unbiased detection.
Metagenomics, meaning sequencing everything.
Pretty much. As sequencing costs continue to plummet, performing shotgun sequencing directly from patient samples is becoming increasingly feasible. Shotgun means you just sequence all the genetic material in a sample, DNA, RNA, whatever's there.
Host DNA, bacterial DNA, viral RNA, everything.
Everything. It's completely unbiased because it doesn't rely on specific primers or probes. It involves sequencing everything present, the host material, DNA from known microbes, commensals or pathogens, and critically, any unknown microbial sequence, however divergent or unexpected it might be.
Can you give us a real world example where metagenomics found something missed by other methods?
Absolutely. There was a powerful case involving an immunocompromised patient who had chronic meningitis, a brain infection, that wasn't responding to treatment and hadn't been diagnosed despite extensive conventional testing.
Okay.
Metagenomic sequencing was performed on their cerebrospinal fluid, and it identified small amounts of DNA from Leptospira, a type of bacterium that can cause meningitis but isn't typically looked for in that context and is hard to culture.
So it was completely unexpected.
Completely unexpected. Retrospective, targeted PCR then confirmed the finding, and the patient recovered fully after receiving appropriate treatment with penicillin G. It was a diagnosis made possible only by the unbiased nature of metagenomics.
Are we seeing this used more routinely now?
It's getting there. Increasingly, whole genome sequencing efforts, including metagenomic approaches, are being applied to other clinical samples like sputum, blood. And recently, a shotgun metagenomic sequencing approach looking at cell-free DNA fragments of DNA circulating in the bloodstream was actually certified for clinical use in the U.S. for detecting pathogens in complex infections. That shows its growing practical application.
But this approach of sequencing everything must also bring unique challenges, right? When you have this massive flood of sequenced data, how do you possibly distinguish the important signal, the pathogen, from all the background noise?
You're absolutely right. That's the major challenge. You need really sophisticated bioinformatics pipelines. First, you have to computationally filter out the overwhelming background of host DNA, which usually makes up the vast majority of the sequences.
Get rid of the human stuff.
Right. Then you compare the remaining microbial sequences to databases of known genomes. But even then, you're left with sequences from commensal microbes, the normal, harmless bacteria and viruses that live on and in us.
So how do you tell if something you find is actually causing the disease versus just being there?
That's the critical step. Distinguishing true pathogens causing the current illness from mere colonizers that might be present but aren't the problem requires very careful interpretation, integrating the sequencing results with the patient's clinical presentation, other lab findings, and sometimes further testing. It's not just about finding a sequence, it's about proving its relevance.
So, moving beyond just identifying known organisms, you mentioned genomic technologies are also being applied to truly discover new pathogens. This sounds like it could modernize Koch's postulates, the old rules for linking a microbe to a disease.
It really does. Koch's postulates, developed back in the 19th century, were a rigorous framework for attributing disease causation to a specific microorganism. A key part was isolating the organism and showing it could cause disease when introduced into a healthy host.
Hard to do if you can't grow it.
Exactly. Current sequencing technologies are ideal for advancing a modern version of these principles. They allow us to identify candidate causal pathogens with unprecedented sensitivity in a completely unbiased way, and crucially, unencumbered by the limitation of whether or not we can culture the organism in the lab.
That's fascinating. For so long, if you couldn't grow it, you essentially couldn't study it properly. But while direct sequencing expands our ability to recognize these associations between a microbe and a disease, what's the necessary caution here?
The caution, and it's a big one, is that critical thinking and further experimentation remain absolutely vital for establishing causality. Just finding a microbe sequence in a sick patient doesn't automatically mean that microbe caused the sickness.
Correlation isn't causation.
Precisely. We can now find many more associations, but proving causality still requires rigorous scientific work, often involving animal models, demonstrating mechanisms of disease, or fulfilling other aspects of modern Koch's postulates. We can't just stop at the sequence.
Virus discovery in particular seems like it's been massively accelerated by these methods.
Yes, definitely. Even early on, Using techniques like high-density microarrays chips containing sequences from diverse known viral families, researchers successfully identified novel viruses. Although somewhat biased towards known viral types, this approach famously contributed to identifying the SARS coronavirus back in 2003.
And its genome was sequenced pretty quickly then, right?
Remarkably quickly for the time. Its full genome was assembled in less than 6 months after the first case was recognized. That was a huge leap.
But now, with next-generation sequencing, the main approach for unbiased pathogen discovery is primarily using metagenomic assembly. Can you walk us through that workflow? How does it go from sample to potentially discovering a new virus?
Okay, metagenomic assembly. First, you take a clinical specimen, maybe respiratory fluid, blood, tissue, and you perform shotgun sequencing. You chop up all the DNA and RNA in that sample into millions of small pieces and sequence them.
Get all the sequences, host, and microbe mixed together.
Right. Then comes the computational part. First, you align all those sequence reads to the host genome, say, the human genome, and you remove them. Filter out the host.
Okay, left with the microbial stuff.
Exactly. Next, you compare those remaining microbial sequences to databases of all known pathogen genomes. Anything that matches a known virus or bacterium gets identified.
But what about the sequences that don't match anything known?
That's the crucial step for discovery. Those remaining unaligned fragments are then taken and subjected to de novo assembly. The software tries to piece these unknown fragments together by finding overlapping ends, building longer and longer sequences, those contigs we talked about earlier.
Creating stretches of the unknown genome.
Precisely. These assembled contigs ideally represent fragments, or maybe even the entire genome, of previously unsequenced organisms. You can then analyze these contigs, compare them to related known organisms to infer their evolutionary position, their phylogeny, and potentially identify them as a new species or strain.
There's a figure in our source material, figure 121.3, that illustrates this nicely, right? Extraction, sequencing, mapping, assembly.
Yes, it lays out that whole workflow visually. DNA extraction, high-throughput sequencing, taxonomic assignment of known reads, then taking the unmapped reads for de novo assembly, and finally phylogenetic comparison to see where the new sequence fits in the tree of life.
And the power of this is just incredible. It identifies not just pathogens you weren't expecting, but potentially entirely undiscovered organisms. The COVID-19 pandemic is perhaps the most dramatic recent example of this in action.
Absolutely. SARS-CoV-2, the virus that causes COVID-19, was identified using exactly this metagenomic sequencing approach within about a month of the first recognized cluster of unusual pneumonia cases in Wuhan.
And the sequencing itself was fast.
Incredibly fast. The sequencing and assembly of its entire genome were completed in just 5 days after getting the samples. And a molecular diagnostic assay, the RT-PCR test, was designed and released literally 1 day later based on that sequence.
Wow. It's truly sobering to think what might have happened if this outbreak occurred even just a decade earlier, before these technologies were so mature and accessible.
It really is. That timeline dramatically illustrates the advancing power and speed of these diagnostic technologies. But it also underscores the pressing need for continued progress, for better global surveillance systems, and for readiness to deploy these tools even faster next time.
Now, beyond these classic infectious diseases like pneumonia or meningitis, genomic discovery is also expanding into conditions maybe not classically thought of as infectious. We already have some well-established links, right, like HPV causing certain cancers.
Yes, exactly. We've known for some time about human papillomavirus, HPV, causing cervical and oropharyngeal cancers, also human herpes virus 8, HHV8, being the cause of Kaposi's sarcoma, and Epstein-Barr virus, EBV, being linked to certain lymphomas. These were groundbreaking discoveries in their time.
What's a more recent, compelling example of this kind of expanded view, linking a microbe to an unexpected condition using genomics?
The very compelling recent example is the link established between Zika virus infection during pregnancy and the devastating birth defect, microcephaly.
How is genomics key there?
Well, epidemiologists noticed that the timing and location of increased Zika virus incidence matched alarming epidemics of microcephaly. But the strongest evidence linking the two came from direct viral detection, using genomic methods, specifically RT-qPCR and even whole genome sequencing, performed on post-mortem fetal brain tissue from affected pregnancies.
So, finding the virus's genetic material right in the damaged tissue.
Exactly. That provided the direct biological evidence needed to build the case for causality, complementing the strong epidemiologic evidence. Both pillars relied heavily on nucleic acid detection and genome sequencing.
So the future potential here seems huge. We're talking unprecedented sensitivity and specificity for identifying foreign nucleic acid sequences, bits of microbial DNA or RNA that might suggest pathogen association in a whole range of conditions,
Yeah.
maybe certain malignancies, chronic inflammatory states, even fevers of unknown origin where conventional tests are negative.
The potential is definitely there. However, and this is a crucial cautionary note we need to keep emphasizing, especially as sensitivity increases. Sequence-based identification alone is not sufficient proof to identify a novel pathogen or link a known microbe to a new disease without fulfilling modern versions of Koch's postulates or providing other strong evidence.
Because we find microbial DNA everywhere now.
Exactly. The increased sensitivity of these methods means we're detecting more and more microbial DNA and RNA in all sorts of samples. This demands even greater rigor in distinguishing true signal, a pathogen actively contributing to disease, from background noise or from harmless commensals or even from contaminants introduced during sample handling or processing.
So again, the mere presence of a microbe sequence is not proof of causality.
Absolutely not. Metagenomic sequencing is fantastic at identifying correlations, but moving beyond correlation to confidently inferring causation requires further targeted methods like specific PCR or hybridization techniques like in situ hybridization to see the microbe in the tissue context in careful clinical and experimental investigation. The bar for claiming a new disease cause remains, and should remain, very high.
This brings us very neatly to a monumental challenge that genomic technologies are now helping us tackle head-on, the cutting edge of resistance battling antibiotic resistance with genomics. This crisis of antibiotic resistance is arguably one of the most pressing global health threats we face today.
It truly is. And traditionally, determining if a bacterium is resistant to an antibiotic has been a slow process. It involves isolating a single colony of the bacteria from a patient sample, growing it up, and then testing its ability to grow in the presence of various concentrations of a drug.
The methods we talked about earlier are culture-based.
Exactly. And this requires, first, that the pathogen is culturable, which isn't always the case. It also requires considerable laboratory infrastructure. And as we've stressed, it involves significant delays. Days for routine bacteria identification and susceptibility testing, and potentially weeks for slow growers like M. tuberculosis.
And these delays have huge clinical implications, forcing doctors into that empirical, broad-spectrum prescribing.
Right. Using those wide net antibiotics because they don't know exactly what they're fighting or, crucially, what it's resistant to. This can lead to treatment failures if the bug is resistant, increased side effects, and it constantly fuels the broader problem of rising resistance globally.
So the idea is that real-time identification and susceptibility profiles would revolutionize patient care.
Absolutely. It would allow doctors to guide therapy precisely right from the start, using the right drug for the right bug.
Yeah.
And it would support the much more judicious use of our precious antibiotics, preserving them for the future.
How does genomics offer a way to accelerate this resistance detection? What's the core principle?
The core principle is shifting from looking at the phenotype, does it grow, to looking at the genotype, does it have the gene for resistance? If a specific resistance-conferring gene or mutation can be identified, it can be targeted for rapid molecular detection using methods like PCR or sequencing.
And you mentioned HIV is kind of the gold standard for this approach.
Yes, in many ways, HIV resistance testing paved the way. Decades of research correlating viral genotypes, the specific sequences of key HIV genes with phenotypic resistance measured in the lab, have clearly delineated the effects of hundreds of mutations in genes like reverse transcriptase, protease, and integrase, which are the targets of antiviral drugs.
Can you give a specific example?
Sure. A single amino acid substitution, called K103N, in the HIV reverse transcriptase enzyme, reliably predicts resistance to a common antiviral drug called efavirenz. It's a very strong predictor.
And this information is widely available.
Yes. Public databases now curate all these known resistance mutations and their effects. So genotypes, the sequence information, are routinely used in clinical practice to predict drug resistance because the phenotypic assays, actually growing the virus with drugs, are far more cumbersome, slow, and expensive.
So it's standard now to sequence the virus before starting treatment in many places.
It is standard of care to perform genotypic resistance testing before initiating antiretroviral therapy in developed countries. This allows treatment to be tailored precisely to the patient's specific viral strain right from the beginning, maximizing the chances of success and minimizing the development of further resistance.
Okay, so that works well for HIV, but you said the challenge for bacterial antibiotic resistance is far more daunting. Why is that? What makes bacteria so much harder?
Several key factors make it orders of magnitude more complex. First, bacteria have much larger and more complex genomes than viruses.
Hmm.
Thousands of genes compared to maybe a dozen in HIV. There are intricate functional interactions between these genes.
And they can share genes, right? That horizontal transfer thing.
That's the other huge factor.
Yeah.
Their capacity for horizontal gene transfer, or HTT. Bacteria can readily share genes, including resistance genes on plasmids and other mobile genetic elements.
Let's just quickly recap those terms for everyone listening to ensure that master-level understanding. Horizontal gene transfer, HGT.
Right. HTT is the transfer of genetic material between organisms other than by normal reproduction, parent to offspring. Bacteria can do this through several mechanisms, like picking up free DNA transformation, direct cell-to-cell contact, conjugation, or via viruses that infect bacteria, transduction.
Okay. And mobile genetic elements.
These are the pieces of DNA that facilitate HTT. They can move around within a genome or be transferred between bacteria. Plasmids, those small circular DNA molecules separate from the main chromosome, are key players. Transposons, or jumping genes, are another type. These elements often carry clusters of genes, frequently including antibiotic resistance genes.
So because of the bigger genomes and all this gene sharing, trying to comprehensively define all the possible ways bacteria can become resistant is vastly more complex than for a relatively simple virus like HIV.
Exactly. It's just a much, much bigger and more dynamic problem space. However, considerable progress has been made in specific cases, particularly where the genetic basis for resistance to a certain drug is constrained to a relatively small, well-defined set of mutations or acquired genes.
Can you give us some of those success stories for bacterial resistance detection using genomics?
Certainly. A classic example is MRSA, methicillin-resistant Staphylococcus aureus. Resistance to methicillin and related antibiotics in Staph aureus is almost always due to the acquisition of a single gene called MECA.
What does MECA do?
It encodes an altered version of a protein called penicillin-binding protein 2A, PBP2A. This altered protein isn't inhibited by methicillin-like antibiotics, allowing the bacteria to survive. And importantly, the MECA gene is usually located within a specific mobile genetic element called a staphylococcal cassette chromosome MEC, or SCC MEC.
So you can test directly for MECA.
Precisely. There are FDA-approved PCR tests that detect the MECA gene, or the SCC MEC element, directly from patient samples or isolators. This can provide an answer in hours, saving days compared to waiting for traditional culture and susceptibility testing. It's a huge time saver for a critical resistance mechanism.
What about other major resistance threats like VRE?
VRE, vancomycin-resistant enterococci, is another good example. High-level vancomycin resistance in enterococci is typically mediated by acquiring specific gene clusters, most commonly VAN-A or VAN-B. Again, these specific genes can be readily detected by PCR assays, reliably indicating resistance.
And for some of those really challenging gram-negative bacteria that are resistant to our last resort antibiotics, the carbapenems, are there genomic tests for those?
Yes, progress is being made there too, although it's more complex. A major mechanism of carbapenem resistance is the production of enzymes called carbapenemases, which break down the antibiotic. Multiplex PCR assays have been developed that can detect the genes encoding the most common types of carbapenemases, like KPC, NDM, OXA48 types, IMP, and VIM.
So finding one of those genes strongly suggests resistance.
It strongly suggests resistance mediated by that specific enzyme, yes. It's a valuable tool for rapid detection and infection control. And one more important example is multidrug-resistant Mycobacterium tuberculosis, MDR or TB.
TB resistance takes weeks to determine traditionally, right?
Exactly. But resistance to rifampin, a cornerstone TB drug, is almost exclusively caused by mutations within a very short specific region of a single gene called RPB, which encodes part of the RNA polymerase enzyme.
So you can just sequence that small region.
Or use a PCR-based assay targeting that region. Automated assays like the GenExpert system do this, detecting both M. tuberculosis DNA and mutations, conferring rifampin resistance directly from sputum samples in just a couple hours.
That's incredible. Hours versus weeks.
It's a transformation. And since rifampin resistance is a strong epidemiological marker for multi-drug resistance in TB, meaning strains resistant to rifampin are very often resistant to isoniazid too, this rapid test provides critical information incredibly quickly, especially in high-burden settings.
But despite these successes, predicting bacterial resistance overall still faces significant ongoing challenges. What makes it still so difficult compared to, say, HIV?
Well, as we touched on, bacteria often evolve multiple diverse resistance mechanisms simultaneously, even to the same antibiotic. Take fluoroquinolones, a common class of antibiotics. Resistance isn't just one gene or mutation.
What kinds of mechanisms?
There are at least 5 distinct modes: reduced drug uptake into the cell, increased pumping of the drug out via efflux pumps, mutations in the drug's target enzymes, DNA gyrase into polymerase effete, enzymatic modification of the drug itself, even proteins that shield the target. And often, a highly resistant strain will have several of these mechanisms working in combination.
Wow, that's complicated.
It is. And consider carbapenem resistance again. While detecting carbapenemose genes is useful, it's not the whole story. Significant resistance can also arise from combinations of other factors, like producing different types of beta-lactamase enzymes, combined with mutations that reduce drug entry through outer membrane porins, or increase efflux pump activity. It's often a complex interplay, not just one gene.
And there are technical challenges too, right? Especially with sequencing those mobile elements like plasmids.
Exactly. Plasmids and transposons, which are often hotspots for accumulating resistance genes, can be technically challenging to sequence and assemble accurately using standard short-read sequencing technologies because they often contain repetitive sequences. Long-read sequencing technologies are improving this situation, but it's still a hurdle.
And it's not always just about the DNA sequence itself.
Correct. Changes in gene expression, how much of a resistance gene is actually turned on or off, can be critical. This might be due to mutations in promoter regions that control the gene, or changes in regulatory genes elsewhere in the genome. Even changes in the copy number of a resistance gene, having multiple copies, can significantly increase the level of resistance. These factors are harder to predict just from the basic DNA sequence.
So predicting resistance when you find a known resistance gene is becoming more feasible.
Yes, the positive predictive value is getting better for many gene-drug combinations.
But predicting susceptibility, saying a bug will be killed by a drug because you don't find any known resistance mechanisms, that's much harder.
That's the crux of it. It's much more difficult to confidently predict susceptibility, to prove the negative. There might always be a novel mechanism you don't know about yet, or a complex interaction you haven't uncovered.
So the ultimate frontier, the Holy Grail, is achieving comprehensive prediction of the resistance phenotype directly from sequence information, getting to that HIV level of confidence for bacteria. But this sounds like a massive needle in a haystack problem.
It truly is a needle in a haystack problem, maybe even finding multiple needles in multiple haystacks simultaneously. There's currently no comprehensive compendium, no complete encyclopedia of all bacterial genetic resistance elements, and, crucially, their complex interactions and their quantitative effects on resistance levels.
And bacteria of the same species can be very different genetically.
Very different. Thousands of genetic differences, SNPs, and gene presence absence variations can exist between clinical isolates of the same bacterial species. But usually only a tiny fraction of those differences actually contribute meaningfully to antibiotic resistance. Finding those critical few among the many is the challenge.
So how are researchers tackling this incredibly complex problem? What are the powerful approaches being used?
The key is combining the vast expansion of microbial sequencing capacity. We're sequencing bacterial genomes on an unprecedented scale now, as you can see in figure 121:1 in the source material, with powerful analytical methods. 2 main approaches are microbial genome-wide association studies, or GWAS, and machine learning algorithms.
Let's break down microbial GDYs again. What is it trying to do?
Microbial GWO is an analytical framework. It takes large collections of bacterial genomes from strains, where we know their phenotypic resistance from traditional lab testing, and it statistically tests for associations between microbial genotypes, specific genes, mutations, genetic variations, and the phenotype of interest, in this case, antibiotic resistance or susceptibility. It's looking for statistically significant correlations across the entire genome.
Like finding genetic markers consistently present in resistant strains, but absent in susceptible ones.
Exactly. And machine learning algorithms can take this even further, learning complex patterns and combinations of genetic features that predict resistance, potentially capturing interactions that simple GWS might miss.
Is there a particular bacterium where this approach has shown a lot of promise?
Tuberculosis serves as an excellent model system for developing and testing these approaches.
Why TB?
Several reasons. It undergoes relatively minimal horizontal gene transfer compared to many other bacteria, and it's primarily restricted to human hosts, which simplifies sampling and evolutionary analysis. Because of this, whole genome sequencing has proven remarkably effective for predicting TB drug susceptibility profiles.
How effective?
So effective that Public Health England in the UK now routinely performs WGS on all new TB isolates, in parallel with traditional phenotypic testing. Some experts believe this is a precursor to potentially replacing much of the slow phenotypic testing with rapid WGS-based prediction in the near future, at least for well-characterized drugs.
That's a major shift. However, you mentioned earlier there's a crucial nuance here between genotype and phenotype, between correlation and causality. It's not always a direct one-to-one translation, is it?
That's a critical point to keep in mind for a deep understanding. Genotype-based prediction methods, especially those derived from GWAs, largely identify correlates genetic markers that are statistically associated with resistance in the population studied. They aren't necessarily the direct causal determinants themselves.
Why does that distinction matter?
Because a correlate might just be linked to the true causal gene, because they happen to be inherited together frequently in that particular bacterial lineage or population. It might not be the gene actually doing the work. In HIV research, a lot of effort went into finding true causal determinants, or reliable surrogates, which were found to be more robust predictors when applied broadly to diverse viral strains.
So a correlative relationship found in one study might not hold up elsewhere.
Exactly. Without a clear mechanistic understanding of why that marker predicts resistance, the relationship might be lineage-specific and less generalizable to other strains or related species. A mutation linked to resistance in E. coli in Europe might not predict resistance in E. coli in Asia, or in a different species like Klebsiella, even if they share a similar gene.
So how do we ensure ongoing vigilance and keep refining these genomic prediction tools as bacteria continue to evolve?
It absolutely requires continuous correlation of genotypic prediction methods with gold standard phenotypic testing. We constantly need to check if our predictions match reality, especially as new resistance mechanisms emerge. You can't just develop a predictive model and assume it will work forever.
But the genomic approach offers advantages even in this ongoing process, right?
Yes, a very important benefit of systematically applying genomics is its ability to systematically identify the outliers, those strains where the genotype prediction doesn't match the phenotype. The ones with unexplained resistance or unexpected susceptibility despite having a known resistance gene.
Why are those outliers important?
Because these discordant strains are where the new discoveries lie. They form the basis for understanding newly emerging resistance mechanisms we didn't know about before. Studying them helps us update our databases, refine our predictive models, and crucially informs the development of new diagnostic tests and even new drugs.
Can you give an example of finding a new mechanism this way?
A major recent example was the discovery of the MCR1 gene. This gene confers resistance to colistin, a last resort antibiotic. It was found on a plasmid, meaning it could be easily transferred between different bacterial species like E. coli and Klebsiella. Its discovery through sequencing efforts, initially in China, raised huge global alarms and highlighted the need for strict surveillance and containment because it represented a readily mobile resistance mechanism to a critical drug.
So while we're making huge strides in predicting resistance from the genome, it's still a complex and evolving picture. Are there other approaches being developed that maybe sidestep the need for perfect genomic prediction, perhaps faster phenotypic tests?
Yes, exactly. Recognizing the current limitations of purely genotypic prediction for some bacteria, researchers are also working on new, rapid, phenotype-based approaches. They're trying to get the functional answer, is it resistant or not, but much faster than traditional culture.
What kinds of approaches?
One interesting idea uses transcriptional profiles, patterns of gene expression, as rapid signatures of susceptibility or resistance.
How does that work?
The concept is that bacteria under lethal stress from an antibiotic they're susceptible to will change their gene expression patterns very differently and much more quickly than resistant bacteria that are happily ignoring the drug. Dying cells are transcriptionally distinct from surviving cells.
So you don't wait for them to die completely, just look at their early response.
Exactly. You expose the bacteria to the antibiotic for maybe just an hour or two, then measure the expression levels of a carefully selected set of key indicator transcripts. Changes in this small set of transcripts can form an expression signature that represents the cellular response, susceptible or resistant, much faster than waiting hours or days for visible growth changes.
Has this shown promise?
Preliminary studies in proof-of-concept systems have shown promise for rapid antibiotic susceptibility identification using this transcriptomic approach. Other rapid phenotypic methods being explored include things like automated microscopy to detect subtle changes in cell shape or division, or even measuring ultrafine mass fluctuations in single cells. It's an active area of research.
And one last point in this section, you mentioned deep sequencing. How does that help us understand resistance within a single patient?
Right, deep sequencing.
Hmm.
This provides unprecedented depth, allowing us to study complex populations of bacteria within a single infection, not just assuming they're all identical clones.
What can we find with that depth?
We can detect heterogeneous subpopulations of the pathogen within that single host. These subpopulations might differ in important ways, including their levels of drug resistance or the presence of specific virulence determinants.
Can you give examples where this diversity matters?
Think about the known diversification of Pseudomonas aeruginosa in the lungs of cystic fibrosis patients over long periods, or the within-host diversity seen in M. tuberculosis during disseminated infection, or the complex quasi-species populations found in chronic viral infections like HIV or hepatitis C.
So it's not always just one bug, but a cloud of related variants.
Often, yes.
All
It's a
this-
cloud of diversity. Deep sequencing allows us to characterize this cloud, identify the major players, but also detect minority variants that might be pre-resistant to a drug or have other advantages.
What's the clinical significance of that diversity?
That's the key question for future research. We still need to fully elucidate the clinical significance of these variable subpopulations. How much diversity is clinically relevant? How does it impact treatment outcome or disease progression? How much diversity gets transmitted? But deep sequencing is providing the tool to start answering these crucial questions, giving us an unprecedented view of the dynamic populations we're actually dealing with in an infection.
Okay, fascinating. Next up, we're going to flip the perspective and explore the host's story. Host-based diagnostics and susceptibility. This is about how genomics is revealing new insights into our own body's response to infection and our individual vulnerabilities.
Right. While pathogen-based diagnostics are obviously central, we've also traditionally relied on looking at the host response. But the markers have been quite limited.
Like serology, which we discussed, has issues with timing and interpretation.
Exactly. And other non-specific biomarkers like ESR, erythrocyte sedimentation rate, or CRP, C-reactive protein, or just white blood cell counts. They tell you inflammation is happening, but not much more specifically. Even newer biomarkers like procalcitonin, which tends to be higher in bacterial infections than viral ones, have somewhat limited versatility.
So they lack the precision needed for many clinical scenarios. Where does genomics come in to help understand the host response better?
A really exciting new direction is focusing on transcriptomic analysis of the host response, specifically looking at the transcriptional profiles, which genes are being turned on or off in circulating white blood cells as potential diagnostic markers for human infectious diseases.
Looking at our own gene expression patterns.
Precisely. Our immune cells change their gene activity dramatically and often quite specifically in response to different types of infections. Can we read that pattern to help diagnose or manage the infection?
Tuberculosis seems like a prime candidate for this, given the difficulty distinguishing active disease from latent infection using pathogen tests alone.
It is, and indeed recent work has shown that the transcriptional profile of circulating white blood cells is markedly different between people with active TB disease and those with latent TB infection, where the bacteria are present but dormant.
How different?
Researchers identified a differential pattern of expression involving nearly 400 transcripts that could clearly distinguish active from latent disease. This pattern seems to be strongly driven by changes in interferon-inducible genes, reflecting a specific type of immune activation happening in active disease.
And has the signature been validated? Does it hold up?
Yes, what's truly insightful is the validation. This transcriptional signature not only distinguished active versus latent TB, but it also successfully differentiated active TB from other inflammatory conditions or other types of infection.
That's powerful discrimination.
It is. And even more, the signature was shown to track treatment responses. The gene expression pattern started to normalize back towards the latent or uninfected state within weeks to months of effective anti-TB therapy.
So it could be used to monitor treatment success.
Potentially, yes. Such a test could become a valuable marker of treatment efficacy in clinical trials for new TB drugs, helping to quickly assess if a drug is working biologically, perhaps long before traditional measures like sputum culture conversion. There's even work distilling this down to maybe just a 3-transcript signature, which shows promise for developing a simpler, more easily deployable assay for clinical use.
Are there broader applications for this host transcriptomic approach beyond TB?
Absolutely. Considerable progress has been made in identifying host transcriptional signatures in blood cells that can distinguish bacterial infections from viral infections, a huge diagnostic challenge, especially in children with fever.
That would be incredibly useful for antibiotic stewardship.
Immensely useful. Other signatures aim to differentiate between different types of bacterial infections, like gram-positive versus gram-negative, or even identify specific inflammatory states. Signatures have also been reported that might help diagnose challenging infections like Lyme disease or predict the severity of influenza.
Is this generalizable across different people?
That's always a key question. But progress is being made in extending these results and validating signatures across different host populations: adults, children, people with varying immune function like HIV patients. The idea is that profiling these host transcriptional dynamics can significantly augment the information we get from pathogen studies, enhancing diagnosis, helping monitor illness progression, and tracking the response to therapy.
What's the next frontier in host transcriptomics?
A major frontier is moving towards single-cell host transcriptional profiling. Instead of looking at the average gene expression across millions of blood cells, you look at individual cells. This allows dissection of complex processes like sepsis, where many different cell types are involved and behaving differently, at a truly granular level. It's computationally intensive, but offers amazing resolution.
They're shifting from the host response to host genetics or underlying DNA. How are genomic approaches being applied to identify host genetic factors that make someone more or less susceptible to getting an infection in the first place?
This is another major area where genomics is having an impact. It's clear that human genetic variation plays a significant role in determining individual susceptibility or resistance to infectious diseases. And genetic loci involved in this often show signs of strong natural selection in regions where certain infections have been endemic for a long time.
Like genes that protect against malaria being more common in malaria-prone areas.
Exactly. By identifying beneficial genetic alleles, specific versions of genes that are more common in survivors or resistant individuals in these settings, researchers can discover genetic markers for susceptibility or resistance.
How could that be used clinically?
In the future, these markers could potentially be translated into diagnostic tests to identify individuals at higher risk, perhaps allowing for targeted preventive strategies or prophylactic interventions before they're even exposed.
And does it also teach us about how the diseases work?
Yes. Critically, these studies often offer deep mechanistic insight into pathogenesis, how the infection causes disease, by highlighting key host pathways involved. This, in turn, can inform the development of new therapeutic methods that target those host pathways.
We knew about some examples even before modern genomics, right? Like the Duffy blood group and malaria.
That's right. The protective effects of being negative for the Duffy blood group antigen against Plasmodium vivax malaria, or having heterozygous sickle cell trait, or other hemoglobin abnormalities, offering protection against severe Plasmodium falciparum malaria. These were classic examples discovered through older methods.
So what does genomics add now?
Genomics allows for a systematic and widespread application of this principle across the entire human genome and across many different populations and diseases. We can now conduct large-scale genome-wide association studies, GWAS, in humans to identify genetic variants associated with increased susceptibility to prevalent global diseases like HIV, tuberculosis, cholera, and many others. It also helps find host genetic factors that predict disease severity, not just susceptibility.
And this is being applied to COVID-19 too.
Very much so. Numerous large-scale GWAS studies are underway globally for COVID-19, trying to understand the host genetic factors that contribute to the incredibly wide variability in outcomes, from people who remain completely asymptomatic to those who develop fatal disease. Identifying these factors is crucial for understanding the disease and potentially finding new treatments.
Okay, let's move on now to shaping the future. Genomics in therapeutics and vaccine development. This is where reading the blueprint helps us design our defenses more effectively.
Right. Genomics has the potential to impact infectious disease therapeutics in two really profound ways. First, as we've discussed, by transforming diagnostic information to influence real-time clinical decision-making about existing therapies. And second, by opening up completely new avenues to understand pathogenesis and therefore develop novel therapeutic agents and vaccines.
Let's talk about that first part, influencing current therapy. We know antibiotic discovery is declining significantly.
It is. There are very few truly new classes of antibiotics in the pipeline. This is partly due to economic disincentives for development, but also because it's just scientifically immensely challenging to develop broad-spectrum drugs that are effective against the extremely diverse range of bacteria we face, especially with rising resistance.
So, how could better genomic diagnostics change the way we use the antibiotics we do have?
This comes back to the potential for a major paradigm shift in antibiotic strategy. If we have rapid diagnostics that provide real-time, reliable antibiotic susceptibility information.
Knowing exactly which drugs will work for that infection.
Exactly. That capability could guide precise antibiotic selection right from the start. This could allow a critical move away from relying so heavily on empirical broad-spectrum drugs and towards using narrow-spectrum agents that target only the specific pathogen identified.
Why is that shift important?
Because using narrow-spectrum drugs whenever possible preserves our precious broad-spectrum antibiotics for when they're truly needed. It reduces the selective pressure driving resistance to those critical drugs, and it minimizes disruption to the patient's beneficial microbiome. It's a cornerstone of good antibiotic stewardship, enabled by better diagnostics.
Genomics also seems to be opening new avenues for personalized medicine in infections, helping us understand why different people respond differently to the same therapy. Can you give us a compelling example of that?
A really powerful example comes from research on tuberculous meningitis, which is a very severe form of TB affecting the brain. Glucocorticoids, or steroids, are often used as an adjunct treatment to reduce inflammation, but their benefit has been inconsistent.
Why the inconsistency?
Researchers discovered that common polymorphisms, natural genetic variations in a human gene called LTA4H, which encodes an enzyme involved in modifying inflammatory molecules called leukotrienes, actually modulated the patient's inflammatory response and their response to steroid treatment.
How so?
Patients who were homozygous for a specific LTA4H allele associated with a high inflammatory response benefited significantly from receiving glucocorticoids. Their outcomes were much better with steroids. However, patients homozygous for a different allele associated with a low inflammatory response were actually harmed by receiving glucocorticoids. Their outcomes were worse.
Wow, so the same drug had opposite effects depending on the patient's genes.
Exactly. This strongly suggests that, in the future, a simple genomic test for the LTA4H genotype could prospectively identify which patients with tuberculous meningitis are likely to benefit from steroids and which might be harmed. It could transform a seemingly standard of care intervention into a precisely tailored, personalized therapy based on the host's genetic makeup, revealing vulnerabilities in the infection process itself.
That's truly personalized medicine in action. Now let's broaden out to genomics and drug and vaccine development more generally. How are these technologies dramatically changing the research process itself, helping us find new ways to fight infections?
Sequencing and related genomic technologies offer several crucial avenues into antimicrobial therapeutic discovery. First, these genome-scale molecular methods allow for the comprehensive identification of all the essential genes encoded by a pathogen,
Essential meaning.
meaning the genes absolutely necessary for the pathogen's survival and replication. If a gene is essential, knocking it out or inhibiting its product should kill the bug. Systematically identifying these essential genes across the entire genome helps us pinpoint potentially critical therapeutic vulnerabilities, new targets for drugs that we might not have considered before.
And how do these methods help us understand how potential new drugs actually work at a molecular level, their mechanism of action?
That's a second major contribution. Genome-scale methodologies offer rapid and unbiased ways to determine the mechanism of action, or MOA, of newly identified antimicrobial compounds. One powerful approach is to evolve resistance to the new compound in the lab, and then perform whole genome sequencing on the resistant mutants. The mutations that arise specifically in the resistant strains often point directly to the drug's target or the pathway it affects. It's a rapid, unbiased way to get clues about the MOA.
What about looking at gene expression again?
Yes, transcriptional profiling is also very useful here. Measuring how global gene expression patterns change after exposure to a compound can provide insights into its MOA. For instance, the transcriptional signature produced by compounds that disrupt the bacterial cell wall is typically very different from the signature of compounds that damage DNA. This can help researchers categorize new hits and prioritize compounds with potentially novel mechanisms of action, avoiding redundant Me Too drugs.
I remember reading about an alternative genomic strategy using RNA interference.
Yes, that was a clever approach used in trypanosomes, the parasites that cause sleeping sickness. Researchers used RNA interference, which can selectively silence genes. followed by targeted sequencing to identify genes that were required for the efficacy of existing antitrypanosomal drugs. This provided new insights into the mechanism of action of drugs that had been used for decades, but whose precise molecular targets were actually unknown.
And the third area you mentioned was vaccine development. How does genomics help there?
Third, sequencing readily identifies the most conserved regions of a pathogen's genome and the corresponding gene products. This information is invaluable for narrowing down the list of potential antigen candidates for vaccine development. You want to target something that doesn't change much between strains.
And this ties into reverse vaccinology.
Exactly. Reverse vaccinology is a fascinating approach enabled by genomics. Instead of starting with the whole pathogen and trying to purify antigens from it, which can be hard, especially for non-culturable bugs.
You start with a genome sequence?
You start with a genome sequence. You use computational tools to predict which genes encode proteins likely to be on the pathogen surface and accessible to the immune system. Then you synthesize those genes, express the proteins recombinantly in the lab, and test them individually for their ability to elicit a protective immune response.
So you work backwards from the genome to the vaccine candidate.
Precisely. This has proved particularly useful for pathogens that are difficult to culture or whose natural antigens are poorly immunogenic. The first licensed vaccine developed using this approach was against Neisseria meningitis serogroup B.
And of course, the most dramatic and timely example of genomics enabling vaccine development was the mRNA vaccines for SARS-CoV-2.
Absolutely. Developed at truly record-breaking speed. That speed was unquestionably accelerated by the rapid public availability of the SARS-CoV-2 genomic sequence data.
How did the sequence guide the design?
Targeting the spike protein was the key decision early on, and that was largely based on its homology, its genetic similarity and structural similarity to the spike proteins from the earlier SARS and MERS coronaviruses, which have been studied for years as potential vaccine targets. Knowing the precise sequence of the SARS-CoV-2 spike allowed for rapid design and optimization of the mRNA constructs.
And these mRNA vaccines were remarkably effective.
Remarkably effective, a true breakthrough. And they offer a potentially very facile platform technology for the future. You can, in theory, quickly swap in mRNA sequences for new viral antigens based on ongoing genomic surveillance of emerging threats.
Genomics has also been employed, you mentioned, to define the impact of vaccines on microbial populations, their epidemiology and ecology. How does sequencing help us understand what happens after a vaccine is widely deployed?
This is crucial for long-term vaccine strategy. Extensive sequencing of influenza viruses, for example, is invaluable for understanding how well the seasonal vaccine is working each year and tracking the global spread of different strains.
What about for other vaccines, like the malaria vaccine?
For the RTS, malaria vaccine, which targets a specific protein called the circumsporozoa protein, targeted sequencing of malaria parasites from vaccinated individuals revealed that the vaccine had reduced efficacy against parasites that had specific mutations in that target protein. That's a key finding for guiding improvements to the vaccine.
And for bacterial vaccines, like the pneumococcal vaccines targeting Streptococcus pneumonia.
Right, the pneumococcal conjugate vaccines, PCV7 and later PCV13, have dramatically reduced invasive pneumococcal disease caused by the specific serotypes included in the vaccines. A huge public health success.
But did the bacteria adapt?
Yes. Sequencing studies clearly documented a phenomenon called serotype replacement. Non-vaccine serotype strains with capsule types not covered by the vaccine increased in prevalence, essentially filling the ecological niche vacated by the vaccine strains.
So the overall disease burden didn't disappear entirely.
It was still greatly reduced, but the types of strains causing the remaining disease shifted. Sequencing also revealed something else interesting. Some of the original vaccine-targeted strains actually persisted by acquiring the genes for a non-vaccine capsule type through recombination, effectively disguising themselves from the vaccine. It highlights the dynamic evolutionary pressure vaccines exert.
What about HPV vaccines? How does genomics play a role there?
HPV vaccines cleverly capitalize on this idea of serotype replacement, in a way. They primarily target the HPV types most strongly associated with causing cervical and other cancers, like types 16 and 18. The aim is not just to protect individuals, but to decrease the overall circulating burden of these most virulent strains in the population, potentially allowing less harmful types to become more common.
So genomic epidemiology helps track what happens to pathogens under the selective pressure of vaccination.
Exactly. It helps us monitor for vaccine escape where the pathogen evolved to evade vaccine-induced immunity and guides the selection of strains for future vaccine updates like we do every year for influenza.
These genomic insights are also leading to exciting new research directions just in understanding basic host-pathogen interactions. How does looking at large-scale gene content, comparing genomes, provide novel insights?
It reveals fascinating things about how pathogens evolve and what makes them pathogenic. For instance, comparing the genome of a standard non-pathogenic lab strain of E. coli, like K12, to a dangerous enterohemorrhagic strain, like the O157.H7 strain, famous for causing foodborne outbreaks.
They're both E. coli, but very different in effect.
Very different. And their genomes can differ by up to 25% in gene content. Much of that difference comes from genes located on mobile genetic elements, pathogenicity islands, that carry virulence factors, toxins, adhesion proteins, etc. That gene content variation dictates pathogenic potential, even within the same nominal species.
And you mentioned Enterococcus earlier, known for drug resistance.
Yes, another striking example. Some hospital-associated strains of Enterococcus faecium, notorious for their multi-drug resistance, have acquired as much as 25% of their entire genome on mobile genetic elements compared to their commensal relatives.
A quarter of their genome is extra.
Essentially, yes. And interestingly, this massive genome expansion through HGT is often associated with the loss of their native CRISPR-Cas systems.
Wait, they lose their defense against foreign DNA. Why?
It seems counterintuitive, but losing CRISPR, which normally protects bacteria against acquiring foreign genetic material like plasmids or phages, ironically makes them more permissive to acquiring new mobile elements, which frequently carry beneficial cargo like antibiotic resistance genes.
So losing their defense helps them gain resistance. That's a fascinating trade-off.
It appears to be. While this might put them at a disadvantage in antibiotic-free environments, these highly resistant CRISPR-deficient strains thrive in hospitals where antibiotic pressure is high, even in the face of our best anti-enterococcal therapies. It shows how environment shapes evolution.
Beyond just sequencing the DNA genome, how is looking at RNA using unbiased whole transcriptome sequencing or RNA-seq changing our understanding of bacterial biology?
RNA-seq is revealing a whole hidden layer of complexity. It's identifying large numbers of previously unexpected regulatory non-coding RNAs in bacteria. These RNAs don't code for proteins but play crucial roles in controlling gene expression, responding to stress, and coordinating behavior.
So they're important regulators we didn't know about.
Many of them, yes. Their prevalence implies significant evolutionary importance and suggests potential new therapeutic avenues that might target these regulatory networks instead of just essential proteins.
And profiling gene expression under different conditions.
That's also incredibly informative. Profiling pathogens using RNA-seq under various relevant conditions like growing in biofilms, interacting in polymicrobial communities, surviving inside host cells, or under stress from antibiotics or nutrient starvation is revealing novel biologic features and vulnerabilities that could be targeted by future therapies. It helps us understand how they actually live and cause disease, moving beyond simple lab culture conditions.
And single-cell transcriptomics is pushing this even further.
Yes, the ability to perform RNA-seq on individual bacterial cells or individual infected host cells is rapidly increasing in feasibility. This is revealing previously unknown heterogeneity cell-to-cell variation in things like intracellular infection outcomes. Not all host cells respond the same way, and not all bacteria within a host cell behave identically. Single-cell approaches let us see that diversity.
So all this genomic information, identifying virulence factors, understanding resistance mechanisms, seeing how pathogens adapt and interact with the host, where does this lead in terms of new drug strategies?
One growing area of interest driven by these insights is the development of anti-virulence drugs.
Antivirulence, meaning they don't kill the bacteria directly.
Exactly. Instead of directly killing the pathogen, which imposes strong selective pressure for resistance, anti-virulence drugs aim to inhibit specific virulence factors, the toxins, adhesion molecules, secretion systems, etc., that the pathogen uses to cause disease.
So you disarm the pathogen rather than kill it.
That's the idea. Make it less harmful, perhaps allowing the host's own immune system to clear it more easily, or making it susceptible to other drugs. The hope is that targeting virulence might impose less pressure for resistance compared to traditional bactericidal or bacteriostatic antibiotics.
And with sequencing becoming cheaper and more widespread, we can now do really large-scale studies linking patient outcomes with both pathogen and host genotypes, right?
Yes. We now have unprecedented statistical power to conduct large studies that associate detailed clinical outcome data with comprehensive pathogen and host genomic data.
What's the ultimate goal there?
The ultimate goal is to reveal critical vulnerabilities in the entire infection process aspects of pathogen biology or host response that are strongly linked to bad outcomes. This could lead to genomic predictors, signatures in the pathogen or host genome that help guide clinical decision-making, predict risk, or tailor therapy more effectively. Imagine being able to sequence the bug in the patient and get a personalized risk score or treatment recommendation. That's the long-term vision.
Okay, this leads us logically into our next section, which is all about tracking the unseen. Genomic epidemiology. This sounds like microbial detective work, but on a regional or even global scale.
It is exactly that. Epidemiologic studies of infectious diseases have always aimed to identify and characterize outbreaks, describe disease patterns and dynamics over time and space, and identify effective interventions.
The classic example everyone learns is John Snow and the cholera outbreak in London, right?
That's the foundational story. In 1854, Snow used meticulous geographic mapping of cholera cases to trace the source of the outbreak to the Broad Street water pump. Famously, removing the pump handle aborted the outbreak, all without even knowing that the causative agent was a bacterium, Vibrio cholerae. That was pure spatial epidemiology.
But now, advances in microbiology and especially genomics have vastly expanded epidemiology's purview. It's not just about mapping cases anymore.
Not at all. Now we can include detailed information about the pathogen itself, its specific strain, its virulence factors, its resistance genes, and analyze the complex relationships between microbial populations and host populations. It adds incredible depth and resolution to the picture.
How do genomic tools provide this higher resolution for tracking?
High-throughput sequencing allows us to describe the diversity within a microbial population, with discrimination down to single nucleotide differences across the entire genome. This gives us far more resolving power than prior methods.
What were the older methods?
Older methods relied on comparing phenotypes, like antibiotic susceptibility patterns, antibiograms, or using genetic markers targeting just a few specific genes, like multi-locus sequence typing, or MLST.
Let's quickly define those for listeners aiming for mastery. MLST.
MLST, multi-locus sequence typing, is a method for typing organisms based on the DNA sequences of fragments from a small pre-specified set of about 7 housekeeping genes involved in basic cell functions. It gives you a broad category, but misses a lot of genomic variation.
And SNPs, single nucleotide polymorphisms.
SNPs are those single-point mutations, a change in just one DNA-based pair. When you compare whole genomes, the number of SNP differences between 2 isolates is a very precise measure of their genetic relatedness. More SNPs means more distantly related. Genomics lets us see all the SNPs across the entire genome, giving incredibly granular detail.
And this granularity is key for tracking transmission.
It's absolutely key. Combined with sophisticated statistical methods grounded in molecular genetics and evolutionary theory, we can translate this fine-grained genetic diversity into detailed descriptions of the origin and history of pathogen spread.
How does that work in practice?
By linking this phylogenetic reconstruction, essentially building highly detailed evolutionary family trees of the pathogens based on their SNP differences with traditional epidemiologic data, like dates, locations, patient contacts, and demographic data, genomic epidemiology allows us to track transmission pathways with remarkable precision, person to person, across geographic boundaries.
So you can infer who likely infected whom.
Yes, you can infer probable transmission chains, identify transmission patterns of both pathogens and their associated resistance elements, and even estimate key outbreak dynamics like the speed of spread.
This sounds particularly powerful for investigating point source outbreaks, say, in a hospital. Can you give us some real-world examples where WGS cracked a case?
Oh, definitely. A really seminal paper back in 2010 used WGS to reconstruct patient-to-patient transmission of MRSA in a hospital in Thailand. They cleverly integrated the estimated rate of mutation accumulation over time with the date samples were collected and the locations of patients within the hospital to build a highly probable transmission map.
Showing exactly how it spread.
Pretty much. Since then, WGS has been used repeatedly in outbreak investigations to define transmission routes and motivate specific interventions. There was another famous case in a neonatal intensive care unit, a baby unit, in Cambridge, UK.
What does WGS show there?
Traditional infection control methods had identified ongoing MRSA cases, but WGS provided much higher resolution. It revealed an unrecognized hidden outbreak caused by one specific MRSA strain circulating amidst a background of other diverse MRSA strains being brought in occasionally. WGS showed transmission occurring not just between babies, but also involving mothers and transmission links back to the community. And crucially, It identified a single healthcare provider who was asymptomatically carrying that specific outbreak strain and unknowingly contributing to its spread.
Pinpointing the source, that's powerful.
Incredibly powerful for guiding targeted infection control. Another example involved an outbreak of carbapenemus producing Klebsiella pneumonia, a highly resistant bug, at the National Institutes of Health NIH Clinical Center in the U.S.
What was the question there?
The key question was whether the multiple cases they were seeing were due to several independent introductions of the resistant bacteria from outside, or if it represented a single introduction followed by undetected nosocomial transmission spread within the hospital.
And WGS could tell the difference.
Yes. By comparing the genomes of the isolates from different patients, WGS showed they were extremely closely related, differing by only a handful of SNPs. This strongly indicated a single introduction event followed by subsequent patient-to-patient transmission within the hospital, proving the latter scenario. This kind of definitive answer helps hospitals understand transmission dynamics and evaluate the effectiveness of their infection control measures. It's also driving research into the cost-effectiveness of using routine genomic surveillance versus non-genomic approaches.
Now, for viral epidemiologic patterns, especially with those rapidly mutating RNA viruses like influenza or coronaviruses, the insights must be even more profound, right? Because they change faster.
They are indeed. The relatively high mutation rate of many RNA viruses, largely due to their error-prone RNA polymerase enzymes that copy their genome, means that genetic changes accumulate much more quickly during an outbreak compared to bacteria.
So you can see the evolution happening almost in real time.
Almost. This rapid accumulation of mutations provides a much finer-grained molecular clock, facilitating even more detailed inferences about transmission pathways and spread dynamics over shorter timescales. These genomic epidemiology tools have been applied very successfully to track outbreaks of yellow fever, mumps, MERS-CoV, SARS-CoV.
And of course, SARS-CoV-2 during the recent pandemic.
Absolutely. The COVID-19 pandemic is arguably the most powerful and visible demonstration to date of genomic epidemiology's central role in tracking the spread of a pathogen, both locally within communities and hospitals and globally across continents. This tracking directly informed public health policy, border controls, and our understanding of how different variants emerged and spread.
What's truly insightful, you mentioned, is how these genomic epidemiology studies are sometimes uncovering completely unexpected transmission events, forcing us to rethink how certain pathogens spread and how we should control them.
Yes, this is where genomics can really challenge long-held assumptions. A great example involves non-tuberculous mycobacteria, specifically Mycobacterium abscessus. This organism can cause severe chronic lung infections, particularly in patients with cystic fibrosis, CF.
And how was it thought to spread?
The prevailing wisdom for decades was that these infections were almost always independently acquired by each patient from environmental sources, like soil or water. Person-to-person transmission was thought to be rare or non-existent.
But genomics showed otherwise.
Dramatically so. Several studies using whole genome sequencing of M. abscessus, isolates from CF patients across different clinics, combined with sophisticated Bayesian modeling to reconstruct transmission trees, revealed that the majority of infections, particularly with certain dominant clones, were actually being transmitted between patients, likely through indirect routes.
Even without direct contact between the patients.
Yes, even without clear, direct epidemiologic links. This strongly suggested transmission might be occurring via contaminated surfaces, fomites, or perhaps even aerosols in shared spaces, challenging the existing infection control guidelines, which were based on the assumption of only environmental acquisition.
That could completely change how CF centers operate.
It has prompted a major reconsideration of infection control practices for M. abscessus in CF clinics worldwide, highlighting the need to investigate these previously discounted modes of transmission. Another clear example, which we touched on earlier, was the M. chimera outbreak after cardiothoracic surgery.
Right, the contaminated heater-cooler units.
Exactly. Finding extremely closely related bacterial isolates, genetically almost identical, in patients from geographically dispersed locations across Europe and the U.S. provided very strong support for a single common point source. This genomic evidence was crucial in eventually tracing the contamination back to the manufacturing process of a specific brand of heater-cooler devices used during open-heart surgery. These kinds of studies continuously advance our fundamental understanding of infection sources and modes of spread, allowing us to establish truly evidence-based strategies for prevention and intervention.
This also raises an important point about the diversity we find within a single infected individual. Traditionally, microbiology often worked by picking a single colony from a culture plate and assuming it was representative of the whole infection.
That was a standard practice, largely out of necessity. But the high-resolution genomic view is revealing that the pathogen population within an individual host, especially in chronic infections or complex situations, can often be a heterogeneous cloud of genetic diversity, not just a single uniform clone.
So that single colony might not tell the whole story.
It might not. It raises fundamental questions about how representative a single isolate truly is. What is the clinical significance of this within host diversity? What evolutionary and ecological processes generate and limit it? How much of this diversity actually gets transmitted during a transmission event? And how does this diversity vary by the specific organism, the type of infection, the host's immune status, and the treatment response?
So these are big, open questions for the field.
They are critical questions. Obtaining comprehensive descriptions of this within host diversity and understanding its population dynamics will be a critically important focus of future investigations. We need to move beyond thinking about infections as monolithic populations and start understanding the complex microbial ecosystems involved both within and between hosts. Genomics is finally giving us the tools to do that.
Okay, let's zoom out even further now and consider the global picture: origins, dynamics, and evolution. This is about using genomics to understand the grand narrative of how pathogens emerge, spread across the planet, and change over time.
Exactly. They were able to place the Haitian strain precisely within the evolutionary tree of the ongoing seventh cholera pandemic. It showed that the seventh pandemic, which originated in South Asia in the 1960s, had spread globally in three major overlapping waves. The 2010 Haiti outbreak isolate was found to be extremely closely related to strains circulating in South Asia around that time, strongly supporting the hypothesis that cholera was inadvertently introduced into Haiti by human travelers, most likely UN peacekeepers arriving from Nepal where a cholera outbreak was occurring. Figure 121.4a in our source material actually illustrates this inferred transmission pathway beautifully.
That's incredible detective work using genomes. What about HIV?
Similarly, detailed genomic analysis of diverse HIV-1 group M strains, that's the dominant group responsible for the global pandemic, combined with historical context, allowed researchers to date the origin of this group back to the 1920s, likely in or around Kinshasa in what is now the Democratic Republic of Congo.
And genomics helped understand its early spread.
Yes. The analysis highlighted how factors like burgeoning colonial-era industry, social disruption, and particularly the development of extensive railroad networks connecting Kinshasa to other parts of Central Africa likely facilitated the rapid geographic spread of the virus from its initial emergence point long before it was recognized as a global pandemic.
And more recently, Ebola and Zika.
Genome sequencing was absolutely invaluable during the 2013-2016 Ebola outbreak in West Africa. It helped researchers understand the geographic spread, track transmission chains, identify factors driving or limiting the outbreak, like demographic, climatic, or administrative boundaries, and monitor viral evolution in real time. Figure 121.4b shows an inferred spread map based on this data. The same applies to understanding the spread of Zika virus across the Americas.
And of course, SARS-CoV-2.
Yes, with SARS-CoV-2, the rapid generation and sharing of tens of thousands and millions of viral genome sequences globally allowed researchers to infer its origin likely in Wuhan around late 2019, and then reconstruct its early, rapid global spread with remarkable detail.
So these efforts really demonstrate the remarkable promise of genome sequencing for outbreak response.
Absolutely. They help elucidate previously hidden origins and paths of disease spread, reveal the forces shaping epidemics, and ultimately can inform more effective intervention strategies. The core insight, as you said, is that genomics provides something like a real-time weather map for infectious diseases.
And this idea leads to the paradigm of real-time genomic epidemiology.
Exactly. This is a potential game changer. Combining portable sequencing platforms that can be deployed closer to the field, mechanisms for rapid data sharing, often through public databases, and open source analytical tools like those showcased on websites like nextstrain.org.
What does nextstrain.org do?
It provides continuously updated phylogenetic trees and geographic visualizations of pathogen spread, like for influenza or SARS-CoV-2, based on publicly shared genome data. It allows anyone to explore the evolution and spread in near real time.
So it creates those weather maps you mentioned.
Precisely. It offers a powerful paradigm for predicting epidemic patterns and guiding public health interventions like vaccine strain selection or travel advisories based on the latest genomic data. This really underscores the critical need for building a new global public health infrastructure, with rapid sequencing and data sharing at its core, enabling early detection and swift informed interventions to prevent the next pandemic. We saw the US CDC's PulseNet system shifting to routine WGS for foodborne pathogens even before COVID, and the pandemic has massively accelerated this trend globally.
Another key aspect of understanding outbreaks is quantifying pathogen transmissibility, how easily it spreads. This helps predict epidemic potential and guides the scale of public health response needed. A critical measure here is the basic reproduction number, R0.
Right. R0, often called R0, represents the average number of secondary infections generated by a single primary infectious case introduced into a completely susceptible population.
And the threshold is R0 is 1.
Exactly. If R0 is greater than 1, the outbreak has the potential to grow into an epidemic. If R0 is less than 1, the outbreak is likely to fizzle out and eventually become extinct on its own. Estimating R0 early is crucial.
How does genomics help estimate R0?
Population genomic analysis, by looking at the patterns of genetic diversity and divergence accumulating over time in the pathogen population, can provide independent estimates of R0 and other key epidemiological parameters. For the 2009 H1N1 influenza pandemic, for example, early population genomic analysis estimated R0 to be around 1.2, which helped confirm traditional epidemiological estimates that were in the range of 1.4 to 1.6. It also helped estimate the origin, date, and location of that pandemic strain.
And similar analyses are being done for SARS-CoV-2.
Continuously. Researchers are integrating genomic data with traditional epidemiological models, data on mitigation efforts, like lockdowns or mask mandates, and even population mobility data to assess the impact of interventions on transmission dynamics in near real time. This kind of integrated analysis, powered by genomics, is expanding our toolkit not just for global pandemics, but potentially even for optimizing infection control strategies within institutions, like figuring out effective return-to-work protocols after an exposure.
Now, beyond just tracking transmission and dynamics, pathogen genomics provides incredibly deep insight into pathogen evolution itself, how microbes adapt and change over time in response to selective pressures from their environment, their host's immune system, or our interventions like drugs and vaccines.
Yes, understanding evolution is key. This has profound implications for clinical decisions, for vaccine development, and for designing sustainable therapeutic strategies.
What are the main mechanisms driving microbial evolution?
Microbes evolve through a variety of mechanisms. There's random mutation, the constant background rate of errors during genome replication. There's gene duplication, insertion or deletion of genetic material, recombination, shuffling existing genetic variation, and of course horizontal gene transfer, which is especially important in bacteria.
Segmented viruses like influenza have another trick, right? Reassortment.
They do. Viruses with segmented genomes, like influenza, which has 8 RNA segments, can undergo reassortment if 2 different strains infect the same host cell. The progeny viruses can get packaged with a mix-and-match combination of segments from both parent viruses.
And that's how pandemic strains can emerge.
It's a major mechanism. The 2009 H1N1 pandemic influenza O virus, for instance, was a complex reassortment containing gene segments derived from avian, swine, and human influenza strains. This potential for novel, potentially dangerous strains to emerge through reassortment is a constant concern, especially with strains like highly pathogenic avian H5N1 influenza. Genomics helps us track this.
Have experiments trying to predict what makes a strain like H5N1 transmissible in mammals?
Yes. Some controversial but informative gain-of-function experiments identified as few as 5 specific mutations in the H5N1 genome that were sufficient to make it transmissible via respiratory droplets between ferrets, which are considered a good animal model for human influenza transmission. This helps understand the potential evolutionary pathways to pandemic emergence.
And what about tracking evolution within a host over time?
Longitudinal studies where pathogens are sequenced repeatedly from the same individual over the course of an infection are very revealing. They show how bacteria adapt, for example, from initial colonization to tissue invasion, or how they adapt to new host environments, or the pressures of antibiotic treatment.
Seasonal influenza is a classic example of rapid evolution, isn't it?
It is. The continuous antigenic evolution of seasonal influenza viruses, driven by immune pressure from the human population, requires frequent updates to the seasonal vaccine formulation. Sequencing studies have been crucial in shedding light on the global spread patterns, identifying the likely geographic origins of new seasonal strains, often Southeast Asia, a hotspot of viral diversity and reassortment, and tracking the emergence of novel antigenic variants.
And we touched on how vaccines themselves drive evolution. What did sequencing show happened after the pneumococcal vaccines were introduced?
As we discussed, PCV7 dramatically reduced disease caused by the 7 vaccine serotypes, but sequencing confirmed the rise of non-vaccine serotypes, serotype replacement. It also showed that some strains originally targeted by the vaccine managed to persist by acquiring, through recombination, the genes for a non-vaccine capsule type, effectively switching their serotype identity to evade the vaccine. This highlighted the evolutionary dynamics under vaccine pressure.
So, genomic epidemiology is crucial for guiding future vaccine selection and predicting potential escape.
Absolutely. By tracking how pathogen populations change in response to vaccination, genomic surveillance helps us anticipate which strains might be emerging threats and informs decisions about which antigens to include in next-generation vaccines.
Finally, can genomics link specific pathogen genotypes to clinical manifestations or patient risk? Can we predict how severe an infection might be based on the bug's genes?
That's a major goal, and we're starting to see progress. A good example comes from Listeria monocytogenes, the foodborne bacterium that can cause serious infections like meningitis and sepsis, especially in vulnerable populations.
What did molecular analysis show there?
By analyzing the genomes of many Listeria isolates from different clinical sources, researchers were able to link specific clonal complexes, groups of closely related strains, to different types of disease and overall virulence. They even identified a novel gene cluster that seems to mediate CNS tropism, the tendency of certain strains to specifically infect the brain and cause meningitis.
So you could potentially identify high-risk strains early.
That's the future vision. Imagine a diagnostic test that not only identifies Listeria, but also tells the clinician if it belongs to a hypervirulent lineage known to cause severe CNS infection. This could help stratify patients, inform treatment decisions, and allocate healthcare resources more effectively, directing intensive monitoring or therapy to those at highest risk based on the pathogen's genomic profile.
This brings us to our final section, global horizons and future frontiers. It seems clear that while these cutting-edge genomic technologies are often developed and first implemented in wealthier countries, their greatest potential impact might be in less developed regions where the burden of infectious diseases is often highest.
That's absolutely true. The potential to leapfrog older, slower diagnostic methods and implement rapid, informative genomic tools could have a transformative effect on health outcomes in resource-limited settings.
How is this globalization of genomics happening?
It's occurring in two main ways. First, through applying advanced sequencing technologies located in resource-rich settings to samples collected globally through collaborative research networks. And second, increasingly, through adapting and importing the technologies themselves for on-site implementation in labs within those regions.
Can you give examples where genomics is already making a difference globally?
We've already seen profound insights from genomic characterization of pathogens responsible for major global health threats like tuberculosis, cholera, Ebola, Zika, and COVID-19, guiding diagnosis, treatment, and infection control efforts worldwide.
The rapid TB test based on rifampin resistance mutations is a key example, right?
Exactly. That automated PCR assay, GenExpert, based on well-defined genetic mutations causing rifampin resistance, has been rolled out widely in parts of Africa and Asia. It allows diagnosis of TB and detection of potential multi-drug resistance within hours directly from a sputum sample instead of the weeks required for traditional culture. That's a massive leap forward, achievable without needing the extensive infrastructure of a full bacteriology lab.
And the high-resolution tracking of epidemics like Ebola, Zika, and COVID-19.
Genomic tracking provided crucial, real-time insights into transmission dynamics that directly informed effective public health responses in affected countries. These efforts almost always involve close collaborations between international groups and local scientists and public health officials. And importantly, they often include significant investment in building local sequencing capacity and bioinformatics expertise, especially in places like sub-Saharan Africa.
So it's building sustainable infrastructure.
Yes, the goal is increasingly to build that local capacity, enabling real-time outbreak recognition and tracking by scientists within the affected regions themselves. This infrastructure then provides broader benefits for diagnosing other endemic diseases and conducting locally relevant research.
The scale of genomic sequencing during the COVID-19 pandemic was unprecedented, wasn't it?
It truly was. The rapid generation and public sharing of over 55,000 SARS-CoV-2 genome sequences within just the first 6 months of the pandemic is a testament to how far global sequencing capacity and data sharing infrastructure had progressed.
And this trend seems poised to continue.
Definitely. As sequencing costs continue to decline, as technologies become more robust and closer to the point of care, and as these advances synergize with improvements in global IT infrastructure and data analysis capabilities, the widespread implementation of state-of-the-art genomic methods in the areas that need them most is becoming increasingly realistic. It holds immense promise for closing critical gaps in global health equity.
The COVID-19 pandemic, of course, served as the ultimate, albeit tragic, showcase for the power and necessity of these genomic tools.
It really did. Its devastating global impact demonstrated the ever-present potential for pandemics in our interconnected world, and it simultaneously highlighted the absolutely central, indispensable role of genomic tools in the response.
From the very beginning, right?
From the very beginning. The rapid discovery of SARS-CoV-2 itself through metagenomic sequencing and the publication of its genome within weeks of the first recognized cases directly enabled the development of the RT-qPCR diagnostic tests that became the global standard.
And it accelerated vaccine development tremendously.
Unquestionably. As we discussed, knowing the genome sequence, particularly the spike protein sequence, and leveraging prior knowledge from SARS and MERS research, allowed for the incredibly rapid design and testing of the mRNA and other vaccines. The progress of the most rapidly developed and validated effective vaccine in human history was undeniably accelerated by genomic technology.
And throughout the pandemic, whole genome sequencing played a massive role.
A huge role. It was critical for tracking outbreaks in specific settings like hospitals, nursing homes, or workplaces, for confirming suspected case clusters, for distinguishing reinfections from prolonged shedding of the virus, for monitoring the spread of the virus through communities, and perhaps most visibly, for tracking the evolution of the pathogen itself.
Tracking the variants.
Exactly. Genomic surveillance was essential for detecting and characterizing the emergence of variants of concern, alpha, delta, omicron, and others that exhibited altered characteristics like increased transmissibility, different disease severity, or the ability to partially evade immunity from prior infection or vaccination. This information was vital for adapting public health strategies and predicting future waves.
And genomics is also helping us understand why COVID-19 outcomes varied so widely between individuals.
Yes, cutting-edge genomic methods, including host genetas looking for human genetic susceptibility factors and single-cell transcriptional profiling of immune responses, are actively contributing to our understanding of the complex factors underlying that wide variability, from completely asymptomatic carriage to severe life-threatening disease and long COVID.
Overall, the pandemic, while devastating, really underscores the essential role of genomics in modern infectious disease management.
It does, both clinically and for public health. And it reveals the urgent, undeniable need for further development and sustained investment in these tools, and for strengthening global surveillance and response infrastructure to be better prepared for the future.
So to summarize this deep dive, it seems clear that genomic technologies are profoundly transforming medicine and infectious diseases. They're illuminating the genetic information that encodes the most fundamental processes of life.
They are dramatically expanding our understanding of the complex interactions of microorganisms with each other, with their human hosts, and with the environment they share.
But there are still barriers, aren't there? Widespread adoption in routine clinical and public health settings isn't universal yet.
No, not yet. Technological hurdles remain, especially around ease of use and integration into existing workflows. Financial barriers are also significant, although costs are decreasing. These factors still slow down the adoption of large-scale pathogen sequencing outside of major academic centers or public health reference labs.
But even so, genomic methodologies have utterly transformed the research landscape already.
Completely. And they are making very meaningful inroads into routine clinical practice right now, as we've discussed with examples like HIV resistance testing, MRSA detection, TB diagnostics, and outbreak investigations.
As even vaster amounts of genomic data are generated, what's needed to handle it all and make sense of it?
That's a huge challenge. We need continued innovation in data storage solutions, further development of powerful and user-friendly bioinformatics tools, ongoing efforts towards standardization of methods and reporting formats so results are comparable, and critically, comprehensive training of the end users, the clinicians, the lab techs, the epidemiologists who need to interpret and act on this complex information.
And there are still fundamental questions to answer.
Yes, critical questions remain about the precise cost effectiveness and clinical utility of WGS for many routine applications, especially in the clinic. We need more rigorous studies demonstrating clear impact on patient outcomes to justify broader implementation in many healthcare systems.
but the momentum seems unstoppable.
It does. The ongoing efforts to overcome these limitations through global scientific collaboration, dedicated teaching and training programs, and continued technological innovation driving down costs are absolutely vital. With the continued rapid advances in genomic technologies and computational analysis, our ability to detect, characterize, treat, monitor, prevent, and control infections has advanced incredibly quickly, and it will continue to do so.
It really feels like it's heralding a new era in medicine.
It is a new era where clinicians are better armed, more precise, and ultimately more effective in their fight against infection and in their core mission to promote human health.
So, what does this all mean for you, our listener? We've journeyed from a time where we were largely, guessing at what caused disease, operating in the dark sometimes, to an era where we can literally read the entire genetic story of a pathogen. We can predict its weaknesses, track its movements across the globe, maybe even anticipate its next evolutionary move.
It's a profound shift in perspective.
This deep dive into microbial genomics really shows us a future where our tools for combating infection are sharper, faster, and vastly more precise than ever before. And hopefully, by going through this in detail, you've gained that comprehensive, master-level understanding we aim for, putting you on par with professionals grasping these concepts.
By exploring these concepts, the shift from phenotype to genotype, the power of NATs and WGS, the challenges of resistance prediction, the insights from host response, and genomic epidemiology. You've truly built a strong foundation, mastering this crucial information.
Now, here's a final provocative thought for you to mull over. As we gain this truly unprecedented ability to detect and understand tiny variations in microbes down to the level of a single host, maybe even within different tissues in that host, what new ethical and practical considerations might arise?
That's a deep question.
Think about patient privacy when we have such detailed pathogen genomic data. Think about how public health interventions might change, potentially becoming much more targeted, maybe even intrusive. Does it change our very definition of health or infection when we can detect minute amounts of microbial DNA everywhere?
How will we balance these powerful individual genomic insights with the broader societal implications, with fairness, with resource allocation? These aren't easy questions.
They aren't. But they're important ones to start thinking about as this technology becomes more pervasive.
We encourage you to continue exploring this fascinating, rapidly evolving interplay between genomics, infectious disease, human health, and the microbial world that surrounds us and defines us. Thanks for joining us on this deep dive.